All right, welcome back to part 3 of Chromosome Organization. Here we are going to talk about the variation in the size of eukaryotic genomes, and we'll also start to talk about repetitive sequences that we find within them. Now, when we go from a bacterial species to eukaryotic species, we see a substantial increase in the amount of DNA. So that might lead you to believe that the amount of DNA correlates with organism complexity. However, when you look at different eukaryotic genomes, they can actually vary substantially in size, and it doesn't necessarily have to do with the complexity of that organism. So, for example, here what we'll see here is that we have two salamander species that, they're both salamanders. They're closely related, but there is a twofold difference in the size of their genome. The difference in size is not because of extra genes. It's because of this repetitive DNA has accumulated and there's more of it. These are regions that do not encode for different proteins. So here, for example, is a chart that shows length of genome size, or the amount of bases present. So here's a million, 10 million, 100 million, so forth. And fungi can range over a long span. Vascular plants are even more spanning. Insects, a lot of variation here. Mollusks, so this is things such as clams and oysters, a little bit shorter. Fish, broad area. Amphibians, broad region. Both reptiles and birds tend to be very narrow in their genome sizes, and we tend to be much smaller as well. Notice that this genome size, plants can have way more genes than we do, or way fewer genes than we do. Recognize that this is a log scale, so each step here is tenfold bigger. So here's in amphibians, those two species that I was referring to, these two types of salamanders. One has twice the amount of DNA as the other, and it has no extra genes. It's just an accumulation of repetitive sequences. All right, so let's take a look at some of these repetitive sequences and think about it. So when we think about sequence complexity, what we're referring to is the number of times a particular sequence appears in the genome. So you might think of this in terms of words. Here we have complexity showing up once in the sentence, but the word "the" shows up twice, right? So there are certain sequences that show up more often than others. And this can be either on a spectrum of unique to non-repetitive, those that are moderately repetitive, and then those that repeat over and over and over again, like saying the over and over again. So let's take a closer look at what these different types of sequences have. So starting off with a unique or non-repetitive, this is where you only find it once or a couple of times in the genome. Remember that each eukaryotic genome has two complete copies of their genome for sexual reproduction. So consequently, there's always going to be at least two sequences. So when we have this includes most of our genes for proteins, and it also has a bunch of the intergenetic regions that are involved in the expression or regulation of the production of messenger RNA of these genes. So in humans, that makes up about 41% of the genome. So here, the vast majority of our genome is not made up of genes. It's not even unique sequences. It's something that's falling into the other categories. So let's move on here to moderately repetitive. So this is when you find a particular sequence that occurs a few hundred times to several thousand times. And this can show up in several different ways. Some of these are actually genes. So some genes that are very, very common and we need a lot of them, we have multiple copies of. Um, so here are RNA, and this is the RNA that makes up ribosomes. And again, ribosomes turn messenger RNA into proteins. We need to do that a lot. So we have multiple copies of those. There are also these proteins called histones. Those are involved in wrapping up DNA. And we need a lot of them to do that. And we also have a lot of variations of those histones that can change how the DNA is wrapped up. We also have sequences that can regulate gene expression. So when you need to turn on a whole set of genes, sometimes we have a particular sequence that will bind a protein that will help do that. So we'll see a lot of those there. We also see things called transposable elements. And these are little sections of DNA that can pick up and move themselves and make copies of themselves. One way of thinking about it is this is kind of the remnants, I'm sorry about that, it's kind of the remnants of previous viruses that have infected our genome and have lost their ability to be pathogenic, but their DNA is still there, and they move up. And oftentimes, we've actually co-opted some of their sequences to do other jobs. We also have very highly repetitive sequences, so this can be found in the tens to thousands to millions of times in the genome, and these tend to be relatively short, so just a few to a couple hundred in length, so a fairly short sequence that repeats over and over and over and over again. And we can see a couple of examples of those. So one is this thing that we call the ALU family in humans. So there's a region of DNA about 300 base pairs long. That sequence occurs over and over and over again, taking up about 10% of our entire genome. These are found fairly regularly, so every 5,000 to 6,000 base pairs. And we'll go into these repetitive sequences a little bit later on. So if you take a look at the entire human genome, about 2% of our DNA encodes for specific genes, so the exon regions of genes, so those pieces of DNA that have codons that encode for particular amino acids and proteins. We also have about 24% that make up regions that include introns, so a lot of the a lot of the DNA has these intron regions, but also the parts in between genes that are involved in regulating how much of that gene is made at particular points in time. And again, these are regions of the genome where other proteins bind to help stimulate that production. And we also have several unique sequences that just aren't in genes. So these are the unique sequences. All of this repetitive DNA makes up the bulk of our genome. And while we'll spend a lot of time going into great detail about how this works, we'll only briefly touch upon this because a lot of it we don't fully understand. Some of it has been described as junk DNA, but we don't necessarily know if that's true. There's many of us that believe that these repetitive sequences play unique roles.